Hi everybody, Natalie here, and today we're going to look at cells, cell parts, and organelles, and try to recognize a few of them, hopefully, in lab under the microscope. All right. Okay, so we'll be learning about first the origination of cell theory, and then we'll try to see the difference between a prokaryotic cell and a eukaryotic cell. Then we'll learn a few organelles, we'll review those organelles, and then I'm going to go into learning even a few more organelles. All right, so it took a lot of work to even figure out that cells were a thing. It took about 300 years of scientific investigation once microscopes started to be developed to have this realization that we are all made of cells and that each cell has very similar characteristics to cells next to us or next to them. Here's a great photograph of two different cell types, the cells lining your bladder and then the cells right under that. And you can see that many of the cells lining your bladder are very similar. They're columnar cells, very similar to each other. Okay. Another interesting thing is that before we understood that bodies, human bodies and animal bodies and plant bodies were made of cells, we didn't really fully understand that cells were super small and that organisms could be single-celled. And so there wasn't previously the idea that doctors should wash their hands. And in fact, some of the first doctors, one of them being Elizabeth Blackwell, who started to insist that doctors wash their hands to avoid spreading cells or disease from one patient to the next, were ostracized. She was ridiculed for her ideas, and she stood up for them. And a few other doctors at the same time period in the mid 1800s were thinking the same things and standing up for those ideas, but it really didn't gain traction for a couple of decades that doctors should actually wash between patients. And this is interesting. The thought was that doctors were so special and good that they couldn't possibly be spreading something bad. Okay. So one of the characters that is instrumental in the theory of cells and the idea of microscopic life was Anton van Leeuwenhoek. He did a lot of work in the 1600s. And interestingly, he developed one of the first really quality lenses because he had a grinding process that was very smooth for his glass lenses that he used on what would be something between basically a magnifying glass and a microscope. He's using it here. So it's a very small handheld device. But he was a cloth merchant, and so he had reason for developing these really precise, clear lenses so that he could see the weave of a cloth very clearly. But after developing these lenses and using them in his trade, he started getting curious and he looked at blood, plaque between his teeth, bacteria in human intestines, the sperm of rabbits, dogs, humans, etc. And his early sketches of, here's sketches of different types of sperm, which is fascinating. Sperm is very, very small, and so he's getting an enormous amount of detail in these pretty rudimentary microscopes. So his sketches really were instrumental in other scientists developing better microscopes and then exploring. Okay, so one of those explorers is Robert Hooke, and he, in fact, gave us the word cell. So previous to Robert Hooke, we didn't have a way to describe the teeny little things that made up bodies. But here's a picture of cork. So cork is a type of tree, and the cork that, might be used to cork a wine bottle or the bottom of shoes or cork boards, is taken from the bark of this tree. And so he's looking at the bark of this tree, and the cells are quite large. And he sees these rectangular shapes. Now, plants have rectangular shapes. Generally, animals don't. But he thought that these rectangular shapes looked kind of like the bedrooms in monasteries. So he called it a cell, because that is the room for the rooms in monasteries. And this is the book that he wrote. So Micrographia, some physiological descriptions of minute bodies. So he's calling microscopic life minute bodies. Minute meaning small, so small bodies. So pretty cool. And you notice he's made by a magnifying glass. So he's using a very precise, very good magnifying glass, not quite a microscope yet. And then it took 200 more years until the 1800s for scientists to build and build and build evidence until they had what we now call the cell theory. And the cell theory, which you should write down, is that all organisms are made of cells. Cells are the basic unit of life, meaning nothing smaller than a cell can be considered life. And then 3, cells come from pre-existing cells that have multiplied. All cells come from cells. This is the classic chicken or the egg thing. And the answer is it's just this cycle that just keeps going back and back and back and back and back. So cell theory does not propose to explain how the first cell on Earth originated, but it does propose how after that first cell on Earth originated all other life. One cell made another cell, made another cell, made another cell. And this here is a bacterial cell budding. It's asexually reproducing, or what we call multiplying, or what we call dividing. I'm so sorry. Apparently, our early scientists were maybe thinking mathematically, but not coordinating their thoughts. So this is one cell that's splitting into 2 cells. Okay, so now we're going to learn the difference between a prokaryotic cell and a eukaryotic cell. All right, so we went over this, that cells are the smallest thing we consider alive, and every form of life on Earth is either a cell or composed of cells. And we also have seen this tree before, where we have our bacteria and archaea on this side of the tree, and our eukarya on this side of the tree. So all eukaryotes here are in one group, and some of them are multicellular. Our animals, fungi, and plants have some that are multicellular. And then our archaea and bacteria are considered a group together. They are called prokaryotes. So when we say the word prokaryote, we mean bacteria or archaea. And our bacteria and archaea are all single-celled. They may grow in colonies where multiple cells grow together, but if I split that colony in 2, I can or scramble it or blend it up. You know, each individual bacteria or each individual archaea will continue to live and exist, and I can't say that that is the case for if you put me in a blender. Okay, so when we group our cells into 2 categories, prokaryotic cells and eukaryotic cells, they look very different. So bacteria and archaea look similar, and so we call them prokaryotes because in many ways it's difficult to tell them apart under a microscope. So our prokaryotic cells are all small. They're all quite simple. They do not have organelles, which are the little organs inside a cell. Little bits, which I will take you through those organelles in a bit here. They first appeared 3.5 billion years ago. So this is the origination of life on planet Earth. So 3.5 billion years ago is the earliest known life that we have found, and that life was prokaryotic. It was a single-celled organism. And so they are all unicellular, single-celled. Uni means one. Okay, and then on our eukaryotic side, our eukaryotic cells are usually larger. They're a bit more complex. They can be up to 10 times larger. They could be 100 times larger than our prokaryotes. They have membrane-enclosed organelles. So that's kind of a lot to unpack. So they have little organelles, and their little organs have a membrane. We can call it a plasma membrane or a cellular membrane, or we could just call it a membrane They're in like little envelopes or sleeves that keep them together. They first appeared 2.1 billion years ago. So they're significantly younger than our prokaryotes. They are unicellular or multicellular, so they can be either. Here is a bacterial cell. So this is much bigger, obviously, than it actually is, and we can see not a lot going on the inside. There is some DNA. There is what we call this flagella, and then here's a plant cell, way more complex. We've got a wall, a nucleus, a vacuole, mitochondria, chloroplast, all sorts of things inside here. So, question for you, are you a prokaryote or eukaryote? So say the word out loud that you are. Eukaryote is what you should have said. Always a good idea, by the way, to practice. I know that it sometimes feels ridiculous, but if you're alone, practice saying prokaryote, eukaryote. These are weird words, so get your mouth used to saying them. Okay, so what are you really made of? Here's a question for you. Did you know that scientists estimate that only maybe 1 in 10 of your cells in your body are actually human? The rest are largely prokaryotic cells. These are generally good bacteria that help us digest food, synthesize vitamins, and protect against disease. So take out your notebook if it's not out already. And I want you to answer this question. How is it possible to have more bacteria cells than human cells? Write your response in your notebook and pause me right here while you're writing. Okay, so hopefully you said something like the cells that are prokaryotic cells are so small that we can't see them. And so we can have more bacteria cells than human cells because our human cells are so huge and our prokaryotic cells are so small that we can fit many prokaryotic cells on, in, or around each human cell without being able to see them. So since a good majority of our bacterial cells actually live in our gut, in our large and small intestine, we actually have a very widely fluctuating number of bacteria cells to human cells. So fun fact, you are more human than bacteria right after you go to the bathroom, if it's number 2. And before you go to the bathroom, you are more bacteria than human. So we get rid of a humongous amount of bacteria when we defecate. And you can look up the stats on this, but it's a good chunk of the weight of our feces that's actually bacteria. So that's fun. Okay, so speaking about bacteria, here's what a typical prokaryotic cell could look like. So I'll start with the black things here, because the black things are structures that we will also see in our eukaryotes. So we have little dots called ribosomes. These build proteins, and these are not visible under a microscope. We have a flagellum, which is a big whipping structure on the outside of the cell, which basically acts like a tail and moves that bacteria through the water. And so in eukaryotes, we also see flagellum. We have a plasma membrane. So this is a membrane that is around the entire outside of our cell, keeping it together, and it regulates the passage of materials from one side of the cell to the outside world. We have a cell wall. So many critters have cell walls. Our plants and our prokaryotes often will have cell walls. And then we have a cytoplasm, which is basically the interior of the cell. So whenever we need to talk about everything to the inside of the plasma membrane, all the liquidy stuff and organelles, or in this case, since prokaryotes don't have organelles, the bits and pieces that are in there, we call that all cytoplasm. All right, and then the special bits that are just things that prokaryotes have. Along with, will have a nucleoid, which is a coiled place where a coiled chunk of DNA. They're DNA will be circular, so like in a ring or a loop, but it will all be mashed like spaghetti all together. And then they'll have plasmids, which are small rings of DNA that float around the inside of the cell. Often a prokaryote will be able to swap these plasmids with other prokaryotes. We have a capsule, which is a sticky outer coat on the outside of the cell wall. It could be filled with toxins to repel our other prokaryotes that might want to eat them. It could be slightly sticky so that it sticks onto surfaces, or oily so that it slides on surfaces. This is variable, but the capsule is basically, a liquidy outer coating. There are pili, which are short projections on the outside of our cell. And prokaryotes have these things called pili that are slightly different from what eukaryotes will have. But the idea is that it's a short projection, not a flagellum, which is a super long projection. But a pili will help a cell move around in space, and even in water. The pili will work very well at organizing movement for the cell. All right. So then if we compare this prokaryote to our eukaryote, the things that we have in common, which you should be writing down, is the plasma membrane on the outside of the cell, the DNA stored either in a nucleoid or a nucleus. We all have ribosomes, which is where protein synthesis occurs. And then everybody's got some type of watery inside that we call a cytoplasm. But our prokaryotic cell doesn't have a nucleus, and it doesn't have organelles. And our eukaryotic cell does have a nucleus, and it does have organelles. All right, so moving to the eukaryotic side, and specifically the animal side. So most animal cells will look very much like this, and that includes our human cells. Our most prominent organelle right in the middle of our cell, which we can often see with a microscope, is called the nucleus. And this is what houses the chromosomes, the DNA, the genetic material of our cells. Right outside of the nucleus that we generally can't see with a microscope is our rough endoplasmic reticulum. This is a folded structure on the outside that produces proteins, and these proteins their instructions for making the proteins come from the nucleus, from the genetic material. So we have these little dots, the ribosomes, found in the rough, embedded in the rough endoplasmic reticulum, making it rough, but also found floating free in the cytoplasm. And they do other parts of protein synthesis. Right outside the RER, or the rough endoplasmic reticulum, we have the SER, or the smooth endoplasmic reticulum. And this is another set of folded membranes, and this synthesizes fats and breaks down toxic substances. So very similar then to our RER, but just dealing with a different type of molecule. Then we have our Golgi apparatus, another folded set of structures where proteins get their final sets of folds so that they can go out into the cell or neighboring areas and function correctly. Okay, moving on, we have one of my favorite organelles called the lysosome. This is the digesting molecule. It digests waste molecules from within the cell. Can eat up whole worn out organelles and spit out basically, atoms, molecules that might be useful to the cell in the future. And then those atoms and molecules get, made into new organelles that are not worn out and will work better. Our cells will have some type of cytoskeleton to maintain the shape and position of the organelles within the cell. It'll have a cytoplasm, so all the stuff, organelles, and liquid on the inside of the cell. We distinguish between cytoplasm and cytosol, for eukaryotic cells. So the cytosol is all the liquid, that's around the organelles, but it doesn't include the organelles, whereas the cytoplasm is all the liquid and all the organelles inside the plasma membrane. All right, then we have, for all of our eukaryotes, we have a mitochondrion. We have mitochondrion, and they are responsible for extracting energy from food molecules and making ATP our energy currency for our cell. So every eukaryote is going to have a mitochondria, or multiple mitochondrion, and they are not usually visible under the microscope. Really, the nucleus here is the only one that I would be able to see under the microscopes at school. Okay, and then we're going to have some type of cell membrane on the outside of it. And so this is going to keep everything inside, but then also control movement of food particles, waste products, and other molecules. All right, so hopefully you got all of that written down. If you didn't, you can pause here and write the rest. Okay, so then within the eukaryotic world, we have two types of cells. We have our animal cells. These are a structure that is very similar for both animals and then for what we call protists, single-celled critters. And our plant cells, though, are really specialized. So they have all the components of a eukaryotic cell, plus they have some other important components, like this, what looks like a swimming pool. This is called a vacuole. We have chloroplasts, which we can often see under the light microscope, and then we'll have some type of cell wall on the outside to give it shape. Okay, so we're going to learn about a couple of organelles, and instead of using cartoons, we're going to see the more real versions of it under the microscope. So we'll look at the nucleus and the chloroplast. So I'm going to go back again here. The chloroplast is right here. So chloroplast, I said, with plants is truly visible. You can generally see it. Okay, so in our eukaryotes, they're all going to have a nucleus. And so let's start at this protist. This is a single-celled eukaryote. It's got a plasma membrane on the outside. I can see that. And then this red blob in the middle, this is the nucleus. I have no idea what this purple thing is, so just ignore it. Then we've got plant cells here. Okay, nice rectangular cells. I can really clearly see the cell walls on these plant cells. And then right here in the middle, I can clearly see a darker section that's the nucleus. And even over here, this cell here is splitting in two, and I can clearly not only see the genetic material, but I can see the chromosomes now condensed. So these, what look like dreadlocks or fingers, these here are, and over here too, this cell is also splitting. These are chromosomes. Okay, so that's the regular plant cells. And then we have skin cell of animal. I believe this is a picture of my inner cheek cell, and like from the inside of my mouth. And so this is our plasma membrane around this whole thing. And then right here in the middle, the darker purple section, this is the nucleus. So nuclei are larger organelles that we can see really clearly. Okay, so each human is going to have 22 pairs of chromosomes plus an additional sex chromosome pair, so 23 total. 46 if you add them all together, assuming that you have 2 copies of each chromosome, which is generally the case, but not always. Okay, so our nucleus will house the chromosomes, and our DNA is wrapped around proteins many, many times until it forms a chromosome. So when our chromosomes are getting ready to condense and then split into 2 different cells, it really folds that DNA molecule very tightly to make a chromosome. Okay. And then we've got the chloroplasts in plant world. So these are the organelle where photosynthesis occurs, which is the process that converts light energy from the sun into sugars. And the sugars then can be processed through the mitochondria, just like in animal bodies, where we will convert our foods into sugars and then process that through our mitochondria. But the cool thing here is that here's our plant cell with our cell wall and plasma membrane. And within it, we can see these green blobs that are chloroplasts. And often they'll be moving around the cell to where the light is the strongest. Okay, so here's another type of microscope. This is an electron microscope. So not something we'll be using in class, but it is something we have on campus at LCC. So chloroplasts here are visible as these blobs again. And you can see the really clear cell wall delineating the borders between cells. This is a little squished because this sample had to be cut in order to be visualized. And so in some places where the cut happened, like this cell here got a little bit squashed, but that's okay. Okay, so our chloroplasts, not ours, sorry, I'm talking as if we're plants. Chloroplasts then have a really cool system of outer membranes and inner membranes to the organelle, and then these stacks of thylakoids where photosynthesis occurs. And photosynthesis is a really cool process, but the equation is basically that it pulls in water and carbon dioxide and light, and then is able to make sugars and oxygen with that. It scrambles those atoms, makes the sugar and the oxygen. Okay. So we have, chloroplasts and mitochondria, and they're both organelles that help provide energy to cells. Chloroplasts then are found in all plant cells. And then mitochondria are found in both plant and animal cells. So plants then have chloroplasts and mitochondria, and animals just have mitochondria. Mitochondria is the organelle of cellular respiration. Now, mitochondria have both an outer membrane and an internal membrane, just like the chloroplasts do. And they use sugar and oxygen that's been breathed in and are able to convert that into carbon dioxide, water, and energy, ATP. So, interestingly, that is the equation that is opposite of photosynthesis. All right. So, let me go back here. So I wanted to just highlight the fact that we have both an outer membrane and an inner membrane for both mitochondria and our chloroplasts. And scientists have a theory for why this is the case. So what they've also found, though, along with an outer and inner membrane, is they've found genetic material like DNA or RNA. I've forgotten which one it is, but some type of genetic material inside of our mitochondria. And so that led them to believe that what could have happened to get the mitochondria and the chloroplast and possibly quite a few other of our membranes that we have in our organelles might have occurred by some type of folding of the membrane of a protist prokaryote. So here's one idea for how maybe a chloroplast or a mitochondria could have come into eukaryotic world. So 2 independent bacteria or prokaryotes are living. One bacteria engulfs or, starts the eating process of the other. Now, when bacteria engulf the other, what ends up happening is the purple bacteria there, the smaller one, is brought in, and that green one sort of takes its membrane and puts it all the way around the purple one. And so then the purple bacteria is brought inside the cell within the membrane of the bigger green bacteria. I know that seems kind of weird. It would be as if we had opened up our mouth and then basically like taken all the skin on the inside of our mouth and just like put it around our food and then brought that down. Like that's not something that works in multicellular world, but this is how single-celled organisms eat. So one bacteria now lives inside the other, but instead of the green bacteria digesting the purple bacteria, it paused, or maybe it couldn't digest it. Maybe there was a mistake, but somehow something happened so that the green bacteria wasn't digested. And if the green bacteria were real, or the purple bacteria wasn't digested, if the purple bacteria was particularly useful, like it was great at making energy or great at photosynthesis, then this would be a mutually beneficial arrangement, because the purple bacteria would get protection from the green one, and the green one would get way more energy or sugar from the purple one. And then since bacteria asexually multiply, and when they asexually multiply, they multiply both their chromosomal material, but also all the other stuff that's in it. They'll split up their ribosomes one part to the next, and then put some plasmids on one side, some plasmids on the other. Maybe at the same time, this purple bacteria is encouraged to, if it's duplicated multiple times inside the green one, we've got a couple purple on one side and a couple purple on the other, and then we pinch off to make two different cells. So then the internal bacteria are then passed from one generation to the next. And this process is a theory we call endosymbiosis. And this is currently the theory that we hold for how mitochondria developed with two membranes and genetic material inside it, and chloroplasts developed as well. So there you go. So make sure you've got endosymbiosis written in your notes, and make sure you've got some sort of definition and maybe some of these pictures written and drawn down for you so that you can come back to this, because endosymbiosis is a really important concept within biology. Okay, so endosymbiosis probably happened a couple of different times. So the first endosymbiotic event would have happened right between the archaea and eukarya zone, so right about here, where a mitochondria was brought in. And then the second and subsequent endosymbiosis events happened on this line within the plants. So probably the plants did this type of endosymbiosis multiple times, because there are a couple of different types of chloroplasts, but there's only one type of mitochondria, and all eukarya share that in common. And within animal world, where we have male and female, the females will give more mitochondria to their offspring than the males do. So we can track movement of humans, for example, or movements of females from one place to the next. And this can be really interesting and fun for tracking the social differences in movement between human groups. So we know that some cultures would have situations where females would leave their homes and move in with their husband's family when they were getting married. We know that there are other cultures where war was a really humongous part of the male lines. And so we have different ways that we can track our female and our male movements within human society, because we have mitochondrial DNA that can suss out female-specific movement from one place to the next. So when we get fossilized evidence, or when we get, frozen humans that have like thawed from permafrost or something, and we look at their genetic material, we also catalog the genetic material in their mitochondria, if that's a sample that's available to us, so that we can track movements of humans. Okay. That was a long aside. Let's talk about some other appendages on cells. So in eukaryotic world, we have something that we have flagella, and that's the same as prokaryotes. So those are the longer whip-like structures on the outside of cells that can move cells through space. And human sperm, really all sperm that I know of, have some type of flagellum. And then in eukaryotic world, we have something very similar to pili, which is cilia. So cilia are projections on the outside of the cell that are small, and they move in a coordinated fashion, like back and forth movement. So they can move a single-celled organism through space, but we also have cilia in our windpipe that can move objects in coordinated effort to move objects up and out if we accidentally swallow something small down the wrong hole. Okay, so let us review. I'm going to show you this slide again, and then I'm going to show you a series of subsequent slides, and I'm going to want you to say whether it's prokaryotic or eukaryotic. So this slide here, you need to write down. So if you need to pause this here, write all this down. And I know that I've gone over it before, so I'm going to shut up. Okay, so we're going to play a game, and I know this is weird in remote land, but what I want you to do is take your notebook and then just write yes or no on it, and we'll sort of grade ourselves, see how well we learned this. So I'm going to ask you a question. I'm going to give you a photograph of some type of cell, and then I'm going to ask you if this is a eukaryote or not. So you'll probably need to have these notes in front of you, and then a little bit of space to say yes or no. Okay, so here's our first slide. Is this a eukaryote? Okay, so the answer is yes. These are plant cells. Plants are eukaryotes, and the way that I know that is that I see clearly that there's a membrane-bound nucleus, so I know immediately we're talking about a eukaryote. But I also can see that there are regular rectangular lines delineating cell walls, and that it's a multicellular organism. So it's multicellular, so definitely a eukaryote. There are no multicellular prokaryotes. And then when I see that it has cell walls that are regular and rectangular, I go, ooh, that's a plant. Plants are eukaryotes. So there you go. You don't see the chloroplasts in this picture. That doesn't mean they're not there. It just means that they're not dyed. But this might be a root picture, in which case we wouldn't see chloroplasts in the roots anyways. Okay, next picture. Is this a eukaryote? Yes or no? Okay, so the answer is no. This is Vibrio cholera. This is the bacteria that causes cholera. You can see a very clear flagella, but just because you see a flagella does not mean it's a prokaryote, because flagella can be on eukaryotes or prokaryotes. So that's not a distinguishing factor. What's distinguishing for me is that when I look inside here, there's not like a big circle thing that's a nucleus. I do see some red dots, but I don't really know what those are. Maybe those are like the shiny outer, like light shining off the outside of the cell, or maybe that's just like little blobs on the inside, but no clear regular organelles inside. So bacteria. Cholera is an awful disease that causes a body to throw up and have massive amounts of, have massive amounts of diarrhea, and you lose an enormous amount of liquid very quickly. And it is fascinating to learn about. Okay, is this a eukaryote? Okay, so the answer here is yes. This is a green alga called Eudorina, and it has chloroplasts. So chloroplasts to, the way, here's my thinking for it. It's unicellular, so automatically I'm not sure if it's a eukaryote or a prokaryote, but I see regular chloroplasts inside. The chloroplasts are green. I don't know of any organelle that would show it clearly under a microscope that's green without any dye, and I can see that there's no dye because nothing else is green here, or nothing else is dyed. So like there's no red outer membrane, which is a dye that we add to our cells. Okay, there are two of them here, but they don't look connected in any way. They're just hanging out together. But because there are chloroplasts, that's an organelle. And an organelle is in eukaryotic land. So green alga, definitely a eukaryote. All right, is this a eukaryote? All right, so this can be a hard one for folks because this is a single-celled thing, and the shape of this looks like a prokaryote, but this is a yes, this is a eukaryote. This is a paramecium, which is a type of protist. It's a lot larger than a prokaryote, which I know isn't useful to you right now because you're not seeing the comparison. But the way I know that this is a single-celled eukaryote is that I see a really clear organelle on the inside of each of them, and that organelle is the nucleus. So I see a nucleus, it is present. Even though it's a single-celled organism, it's definitely a eukaryote because it has that. But if I personally were looking at this under the microscope, I would also know that it was a protist or a single-celled eukaryote just because it's so much larger than a single-celled eukaryote. Did I say that? Single-celled eukaryote, and I know that it's a eukaryote because it's so much larger than a prokaryote, which would be probably the size of this little tiny dot. Okay, and then is this a eukaryote? All right, so this is a trick question. This is both. This is this big thing here in the middle. This is a eukaryote with a nucleus. So cell membrane, nucleus in the middle. And then all of these little dark purple blobs, these are bacteria. So for context, this is a cheek cell, so scraped from the inside of someone's cheek and then put into water under the microscope. And so we can see this is probably saliva, I would guess. And then all these oral bacteria, which we know that our mouth are completely full of bacteria, both good bacteria and problematic bacteria. And here's a big old chunk right over here of bacteria. And then we've got our big cheek cell. So really humongous difference in size there that we can see. All right. So with that, I will pause this lecture and follow up in the next lecture for this week. So have a great day.